Dr. Jeffrey Petruska, part of the University of Louisville Department of Anatomical Sciences and Neurobiology, and also the Kentucky Spinal Cord Injury Research Center. Axon growth is a very important part of neurological recovery, particularly in traumatic situations such as traumatic brain injury. And a lot of work has gone into trying to understand the genes and proteins, molecules involved in controlling axon growth after injury. So that's when an axon is cut and it regrows. We call that regeneration. We know that there's another process that happens, that neurons that are not injured also undergo a growth, and in some cases that can compensate for lost functions. Well, unfortunately, in other cases, that axon growth establishes new circuits in the spinal cord of the brain that end up being somewhat pathologic or problematic, but it's still the same process. And that process is not very well understood. It was actually when I was with the Christopher and Dana Reeve Foundation's Research Consortium on Spinal Cord Injury, which is a magnificent entity. And that group supported a proposal I had to establish this model of collateral sprouting. And we did a microarray experiment. The goal there was to identify which genes were changed in this process of collateral sprouting. I began my own lab when I came here to Louisville. I recruited an outstanding postdoctoral fellow, Ben Harrison, very well trained in molecular biology, skills I needed to help me with this project.
Yeah, I was originally attracted to the project because Jeff had outlined a really good way to analyze two different growth modes of plasticity. And he was working on the idea, the hypothesis, that these two different growth modes would be very distinct to one another with respect to gene expression, with respect to the transcription that's going on during the plasticity. And so when I joined, I thought, well, we could put this idea to the test using bioinformatics. So we were talking to the bioinformatics groups over here at KBRIN, and we obtained funds from them because they saw that what we were doing was pretty exciting as well. They gave us a grant to do what's called a next generation sequencing project, where we sequence all of the genes that are being expressed in plasticity. And we analysed those expression profiles, and we found that there are a lot of these segments of genes that are being regulated during plasticity. And I've been working with KBRIN on that data set. And it's really great to be working with Eric at the bioinformatics group here. He's one of the first guys actually to do groundbreaking work on novel segments of genes, so it was perfect.
And we started to identify genes that looked like they were going to be involved in this collateral sprouting process. We started investigating them, and they turned out really well. And we asked the question, are they also participating in regeneration, which is growth after injury? And they were not. This was very compelling evidence for us that there might actually be two different genetic programs controlling axon growth in the adult. I was fortunate enough to recently be awarded an NIH, they call it an R01 award, which is a 5-year award, a little over $200,000 a year. We proposed a set of experiments that would help answer questions which the NIH and the reviewers felt were important enough and that we could do them. And the KBRIN support, both in terms of expertise, material, time, was absolutely vital for receiving this federal grant and a number of other smaller grants and publishing manuscripts that are being cited. And all this work really comes down to a supportive environment from the Spinal Cord Injury Research Center and the KBRIN.